We're going to start. Good afternoon to everybody. We are starting the session of Evolutionary Ecology, and we have a pretty tight time schedule, so I will directly introduce our first speaker, Jonathan Silverton, who will be talking about evolutionary ecology of senescence.
Right, I'm going to have trouble. Thank you, Jesus. End the meeting on a downer, really. We all die, and the rate of mortality increases inexorably with age. This is what happens in the human population. This is actually human females, but the male pattern is pretty similar. So on the vertical axis, you've got death rate per year. On a log axis, more bad news. And on the vertical axis, you've got age. And by the time you reach 100, if you were lucky enough to do that, the chance of making 101 is only about 50%. So this is actually pretty universal among all organisms, with the exception of those that reproduce clonally. So here's bristlecone pine, looking pretty ancient and senescent, but it is actually very old. And here are some annual plants. These happen to be in South Africa, but in any arid environment, you'll find plants that actually die after 1 year. So although senescence itself is universal, the rate at which senescence occurs is extremely variable indeed, from very slow to horribly quick. And the question is, why do different species age at different rates? So I see Hal Caswell has just walked in, and he knows the answer to this. So I'm going to hand over to you. But this is G.C. Williams, looking fairly old. He died a while ago. And you'll see this is a recurring theme. And he proposed, I'm not sure it was the very first proposal, but anyway, so one that has been interesting people for a long time is that the reason different species senesce at different rates is because the adult death rates are different. This is caused by differences in exposure to environmental mortality. And that a species in which the adult death rate is low will senesce slowly, and a species in which the adult death rate is high will senesce quickly. And he says, according to the theory, that's his theory, death rates prior to maturation, so what happens before puberty, if you like, has no influence on the evolution of senescence. From about 1992, this has come to be called extrinsic mortality. So from here on, I'm going to describe this hypothesis as the extrinsic mortality hypothesis. And it's one that has actually grown in popularity since Abrams in '92 described the Williams hypothesis as relating to extrinsic mortality. The idea is that extrinsic mortality is not dependent on age. It's just sort of random mortality that comes from the environment. That's the idea. And the more of that there is affecting adults, then the faster senescence will occur under evolution. So these are points in the Web of Science representing the number of publications per year. It's not a huge number that actually mention extrinsic mortality and senescence. So you see on the left there, 1992, when the term was introduced, although the idea, as I've explained to you, was introduced earlier, and roughly over time you get more and more papers that are saying we're going to test Williams' hypothesis that extrinsic mortality accounts for the rate of senescence. And this has increased and increased despite the fact that Abrams himself in 93, Howe in 2007, and Vensic et al., and I think Howe was a co-author of that in 2016, have pointed out that the hypothesis, that Williams' hypothesis, is actually flawed. It's wrong. This idea has been tested and tested and tested, and people said, oh, you know, my data agrees or my data doesn't agree. And they've treated this hypothesis as though it's actually sound. So what's the alternative to the hypothesis? Well, Williams said that, and Hamilton said that. Well, not exactly. That's actually a slight reformulation of what he said. And I've been to several talks this morning, and everybody's promised there will be no equations. So I can't make you that promise. But what I can do is actually try and break it down to make it more palatable. So I had to do this for my own use anyway. So on the left-hand side, we've got the force of selection against mortality with age. Okay? That's beta. And those other things is essentially index it by the age and fitness. And on the right-hand side, we've got the sum over the rest of the lifespan, so from age A onwards, of future reproduction discounted by the rate of population increase. The point here being that if you reproduce young, you actually make a bigger contribution to the finite rate of increase of the population than if you reproduce old, just because you've got more time. You're in at the beginning, right? It's the compound interest effect, essentially. Now, so here we've got an age-specific theory of how fitness varies with, how selection on mortality varies with age, whereas Williams' one is essentially age-independent. He says that. And so now what we can do is actually add in a component for extrinsic mortality and see what that does to beta on the left-hand side. So the way you do that, basically, is to the term for discounting the population rate of growth is you add a term EXD. So D is the mortality, the extra mortality that's coming from the environment. And that's the same at every age. D is the same value. X is the age. And then you have to do the same thing also for LX. So LX is the per year survival rate. Again, it's age specific. And what you find is that actually those two terms cancel out. So when you do that, you basically end up, when you add extrinsic mortality to Hamilton's formula, you end up with the same formulation. Extrinsic mortality makes no difference to the selection on mortality. Another way of thinking about this is that actually by definition, D affects all age classes equally. So it doesn't, as a consequence, it doesn't change the age structure, and so it doesn't change the selection gradient with respect to age. It just doesn't make any difference. Thank you. Right, so this is, as I've already pointed out, several people have already pointed this out. So this isn't new, although it does appear that one needs to keep repeating it, so that's what I've done. And why has this flawed hypothesis of Williams been so successful? Why is it actually grown in popularity over time when it should have just disappeared? One reason I'm standing up here and talking about this is because I made the same mistake, I have to say. And in just talking about it with a colleague of mine in Edinburgh, came to what I'm now telling you. So, and one of the things that I found really beguiling about the hypothesis was it does appear to account for the variation in senescence that we see around us. So Williams himself said, well, this can account for why birds actually live longer than terrestrial animals of the same body weight. And actually, it accounts for a whole, if you believe it, I'm telling you shouldn't, that it also would appear to account for why bats live much longer than terrestrial mammals of the same body weight. Arboreal versus terrestrial mammals, fossorial things like moles versus terrestrial vertebrates, poisonous versus, like they all seem to be telling us that something about resisting extrinsic mortality, which delays senescence. So the quick answer is, and this is sort of cop-out, and this is where I also subscribe to the post-math era, after the party last night, is that if you add in selection for late life survival into Hamilton's, so if you look at the effect of extrinsic mortality on the Hamilton formula, you can actually come up with any prediction you like depending on where the mortality is applied. So once you introduce age specificity, there's no longer any simple rule. And the simple idea that Williams came up with, which is all depends on adult mortality, extrinsic mortality, it just doesn't apply. So we can potentially explain, I'm sure we can, the patterns I've mentioned to you down there in the red, but we're going to have to be much more specific about what it is that's selecting for these patterns. Williams was right in perceiving something to be explained and some interesting patterns, but his actual explanation appears to be mathematically flawed. So here are just three examples taken from that graph of dots. So these are three of the dots of studies that have looked at these kind of things. And what you typically find is they don't all support Williams, and we do need more information in most cases to actually explain the results. So here's a study of Daphnia in water bodies. Some are temporary, some are permanent. And actually, sure enough, this would appear to agree with Williams. But actually, the chances are that, when a pond dries up, the mortality isn't being added to the population age non-specifically. It's actually affecting adults. It's affecting a particular cohort in the population. So it's actually not extrinsic mortality as defined by Williams at all. So the agreement with Williams is illusory, really. And here's an example where you've got the presence and absence of predatory fish. The authors said this is actually, extrinsic mortality in one pond and not the other. Turns out there's no difference in the lifespan between these different lakes. And the explanation would be, well, it's not actually affecting age-specific mortality. It may be changing the mortality in a non-age-specific way. And so on. There's another example. This is probably the best known one, a huge experiment by Stephen Stearns. And again, he said he was applying extrinsic mortality, but actually he was just removing adults. So there are larvae in the life cycle as well, and they weren't affected. So it's actually very difficult to test this idea, and most of the experiments that have been done on this could be interpreted correctly. So let me just summarize. So extrinsic mortality does not select for senescence. Williams was wrong. And juvenile mortality does matter. He said it didn't, but it certainly does, because the age-specific nature of the mortality is what counts. And you can actually get a selection for late life survival, in other words, postponed senescence, by a variety of different routes, depending upon exactly when mortality occurs in the life cycle and how bad it is. And finally, there is no simple rule. And if you want to read the book in which I got all this wrong, it's there. There are good bits too. Thank you.
Thank you very much. We have time for some questions.
Thanks for talking. trying to develop a unified theory of senescence is not possible? Or
No, not at all. It's just not Williams' theory. It's a bit more complicated.
Just as a comment, one of the things that's another interesting aspect of Williams' original work, and Hamilton's for that matter, is that it was explicitly designed as theory for age-classified populations in constant, unchanging environments with no density effects.
Right.
That's the demographic model that they used. And the result that age-independent extrinsic mortality has no effect holds also for stochastically fluctuating environments, holds for stage-classified as opposed to age-classified life histories, and very special case, but holds for a special case of density-dependent life cycles as well. So it is really you need to pay attention to the age specificity, regardless of what kind of environment you're looking at the organisms in. But it's not, as you said, it's not impossible. You just have to.
Yes. Thank you. Yes.
Okay, thank you very much.
Thanks.
Thank you. So our next speaker, I haven't seen you, but maybe, yeah, she's here. Morgan Tidier, who's going to talk about evolutionary ecology of senescence.
So good afternoon. So I will present you a last result concerning the survival benefits for mammals living in captivity. From the last century, zoo aims to achieve other goals than only recreational purpose, and they try to have also a role in educational, in wildlife, but also they invest highly in conservation and research programs. And 30 years ago, the World Association of Zoo and Aquarium decided to start emphasizing and sharing husbandry standards in order to improve captive conditions, but also animal welfare. And one of the indicators of this welfare in captivity is the survival. But survival cannot be studied at individuals, but at populational level. Indeed, in his life, an individual can die only once. So if you consider several individuals of same populations, you can have the distribution of the age at death. And from this distribution, you can draw the mortality pattern of this population. So for instance, for this wild population of Lyon, you can observe three main phases. So the juvenile phase with a high mortality rate, the primate phase after the age of sexual maturity with a very low mortality rate, and after you can observe a progressive increase of the mortality rate with age. And this defines the actuarial senescence part of this population. Actuarial senescence can be defined with two key metrics, which are the age at the onset of senescence and the rate of senescence. And we can also add the survival metrics, which is the longevity, defined here as the age at which 90% of the population is dead. So this is for the wild population, but if we draw the mortality pattern for the captive population, because of the highly protected environment provided by zoo with no starvation, no predation, treatment against most of diseases, but also few or no competition for resources, we expect that for captive population, there is a delayed onset of senescence. They senesce slower and then live longer than their wild counterpart. For now, most of studies dealing with these issues have been species-specific and provided contrasting results according to the species considered. For instance, in these two closely related species, we can observe different effects of this of life in captivity. So for the red deer, you have a positive effect on their life expectancy, but for the roe deer, there is a negative effect. So the challenge is to understand what factor could explain this variability of answer to live in captivity according to the species. So to my knowledge, 2 studies have tried to compare captive and wild population of mammal, and they both highlight a decrease of rate of senescence in captivity. But they considered only the rate of senescence and not the onset, and they include less than 25 species. So using demographic data from 59 mammalian species, including 8 different orders, we tried to find factors which can explain the variability of benefit of living in captivity. So for wildlife tables, I used data come from literature, while for zoo life tables, data come from the Species 360 database. So for each sex of captive and wild population of each species, I obtained the onset of senescence, the rate of senescence, and the longevity. And analyses are corrected for the phylogenetic relationship among species. So to read the results, you have the zoo value in Y and the wild value in x-axis. The gray dotted line represents when the zoo value is equal to the wild value. So above this gray dotted line, you have when the species have a benefit from captivity, and below when the species have negative effect from captivity. So for longevity, we observe that for males, more than 80% of our species considered live longer in captivity than in the wild. So captivity seems to extend longevity, probably because increasing age-specific survival. But you can see that the benefit is not the same for all species. There is a high benefit for short-lived species. The benefit is less important for species living 10 years in the wild. And for species living more than 20 years in the wild, such as primates, most of primates, we have a negative effect of captivity on their longevity. The same result is observed for females, and it seems that the species space of life determines the potential of benefits to live in captivity for mammals, with short-lived species benefiting more of captive condition than long-lived species. Similarly, for the onset of senescence, we observe that species with an early onset of senescence delay more the onset of senescence in captivity than species with an onset of senescence around 10 years of life in the wild. So once again, it seems that the species space of life determines the potential benefit of to live in captivity in terms of senescence. And finally, for the rate of senescence, so plots have to be read in the inverse, with here a negative effect of live in captivity and here a positive effect. Okay. And once again, we can see that for a very long-lived species or for slow species with a slow rate of senescence in the wild, there is, they tend to increase their rate of senescence in captivity, while for most of these species, this seems to have a lower rate of senescence in captivity compared to the wild. So once again, species space of life seems to impact the influence of to benefit of captivity in terms of senescence. So we highlight that we confirm the typical belief that captive mammals perform better in captivity than their wild counterpart. And the conditions offered by captivity allows to delay and slow down senescence and then to live longer. But we saw that this result is not true for all mammalian species. And we saw that for very long-lived species, it seems to have a negative effect of captive life on their survival and longevity. And this result is particularly important for conservation issue because long-lived species are particularly threatened by extinction in the wild. So to explain this result, there is the first explanation could be that long-lived species already have very low mortality rate in the wild, so it's maybe not possible to decrease again their mortality rate in captivity. A second possibility could be that long-lived species are often social species, and in captivity, maybe important social relationships are not respected due to management reasons, and this can be associated to stress, which is detrimental for the organism. And finally, management improvement began in 1985. So maybe that for very long-lived species, this improvement of captive condition is not yet observable on their survival and longevity. So given the huge biodiversity loss observed, it's important to consider all possible tools to avoid to reach a point of no return concerning biodiversity. And the zoo and aquarium can provide some help to reaching this goal. And they can help at different levels. So for instance, helping to research programs to a better understanding of factors influencing survival, senescence, and longevity of threatened species, and then maybe improve conservation programs. They can also help maintaining self-sustaining populations to reinforce and reestablish wild populations. And finally, they can also help giving priorities to threatened species and releasing captive pressure exerted on species not directly threatened by habitat loss, such as marine mammals. So the important message of this result is that the survival benefits from captivity are related to the species space of life for mammalian species. So I'd like to thank all of my collaborators and supervisors and all of you for your attention. Thank you.
Thank you very much. Is there any question?
So really interesting comparisons. One of the things about large, slow species is, I think, that when they're subject to density effects in the wild, they tend to protect their longevity and do that by either skipping reproduction or extending interbirth intervals and allowing their fertility to take the impact. That would, if that's the case, and if the wild mortality schedules that you have come from populations where that kind of thing is going on, their mortality would already be relatively low, their survival would be relatively high, and therefore a comparison with the zoo wouldn't have any room to increase it. Do you think that that choice of a strategy, whether you respond to stress by protecting your longevity and sacrificing reproduction, or the other way around, let your longevity suffer, would have some explanation for why you get the body size effects that you do?
I don't understand the question.
So, as an explanation for why the long-lived species don't seem to benefit in survival, in the wild, long-lived species tend to protect their survival if they get stressed because of lack of resources. They want to keep their survival high, and they sacrifice instead reproduction. So that would mean that measured wild mortality schedules already have high survival. So do you think that might be why those species tend to not show a benefit from living in the zoo conditions?
I think that if very long-lived species, very large species, do not benefit from captivity, it's because they already have this strategy to have very low mortality because of they choose to increase their longevity despite their reproduction. But I think that it's also because in captivity there is a lot of stress with the species. Large species need more space, and it's difficult to provide all of this space for them, such as elephants, for instance. So it's really difficult to decrease their mortality rate because it's already very low. But in addition, there is more stress because for very short-lived species, for instance, they do not live very long. So maybe there is no accumulation of stress all of their life. So I think there's a combination of things to explain this, that there is no decrease of mortality rate in captivity.
Very quick, yeah.
If long-lived species could live longer in zoos, if you could remove the stress, would there be any conservation gain? Because often they're not reproductive and they're lacking in ability.
From my point of view, I don't think that keep very long-lived species in captivity is the conservation, is the way to conserve them. I think that for very long-lived species, as it's really difficult to improve their captive conditions, it's maybe more important to concentrate efforts on their in situ conservation. Because effectively, if we increase their longevity but we are not able to increase their reproduction, I think it's not very useful.
Okay, thank you very much. Our next speaker is Ian Staub, who will be talking about consequences of aging for population dynamics.
Okay, does this thing work? Yeah. And the light, yeah, okay. Hi everybody, I'm Ian. I'm based at the Max Planck Author Center for Biodemography of Aging, which is also called MASO. And I want to start off by just saying that obviously this work is done with collaborators, especially my supervisor, Owen Jones, and was funded, at least in part, by the Max Planck International Research Network on Aging. Okay, I'm going to talk about consequences of aging for population dynamics across the tree of plant life. I forgot to put the word plant in my PES abstract, but we're talking about aging and population dynamics in plants today. So we'll dive right in. This is the tree of life, okay? Across the tree of life, there are loads and loads and loads of different ways of living. And there's loads and loads and loads of different ways of living across your age trajectory in life. So we can pick out some examples, some plant examples. This here is quaking aspen, Populus trianguloides, and quaking aspen lives for a really long time. Each one of these trees is a clone, and they're all connected underground by a big root system. And they reckon that there's stands of these trees that live for 80,000 years, so it's probably the oldest organism on the planet. We can go to another part of the tree of life here, and this is Villellas, Arabidopsis thaliana. You should be familiar with it because it's a very popular study organism. And one of the reasons that it's a popular study organism is because it has an extremely fast life history. So it can go from being a seed and germinating to setting its own seed in 6 weeks, which means that in the lab you can grow lots and lots of generations very quickly. And once it sets seed, it dies. So its mortality is very concentrated towards the end of its lifespan. Go to another part of the tree of life here, and this is the talipop palm. And it has some similarities to Villellas because what it does, it has the largest inflorescence of any plant. So this is the inflorescence. And the top of this tree is this 6 to 8 meter bunch of flowers. But the talipop palm also dies as soon as it's reproduced. So it lives for maybe 30, 50, 80 years and then it dies. So again, it has its death concentrated towards the end of the life cycle. But then there are other plants which do very different things. So Rhododendron maxima, the great rhododendron, is very different to the talipop palm because its mortality is spread quite evenly across the life cycle. So it doesn't matter what age you are, if you're a great rhododendron, you have a sort of a similar chance of dying. So be pleased that's the end of all my plant examples, but just to get you excited about plants. So age patterns of death and birth vary among organisms. This is just a very true thing. Organisms live in different ways. And we can measure that now because we have really cool data. So the compadre plant matrix database holds information on survival, growth, fecundity across age or size or stage for thousands of models for hundreds of species. And if you want to know what a model looks like, you can give a bit of visualization. So here is a life cycle. We have small individuals growing into larger individuals, and at each stage of your life you have probability of staying the same life stage that you are now, a probability of advancing, perhaps a probability of regressing, going back. So if you're a big tree, you could be chopped down in size, or you could be grazed on. So you could go back through the life cycle, and you also have reproduction here as well. So this is a sort of a visualization of what's in the compadre matrix database. So we have information implicit in this, because what we can do is add rates to each of these life cycle transitions. And implicit in this is death. So we know what the survival of things are, which means we know how many individuals die. And what we can do, even though a lot of these things are based on size or based on stage, is we can kind of simulate movement of individuals through the life cycle stages. And then we get the measurement of over age, your survival. Simple survival curve, it would look something like this, or something like this, or something like this. And information in here tells you about what your maximum life expectancy is and where your death is spread across the life cycle. So these should be familiar to many of you, and we can get these for models in compadre. And I want to talk to you about how you measure aging, and I want to talk to you about the pace and shape framework. So this is coined by Anessa Baldish, who is in our department at Maxo. And pace and shape says that there are two kind of orthogonal dimensions in your lifespan. There's your pace of life, which is measured along the age axis. And that could be something like life expectancy. So for the rest of this talk, when I talk about pace of life, I'll be talking about life expectancy at birth. Okay, so this is a measure of aging, so just in the sense of how long you live. And then there's another axis, which is dimensionless, which is the shape of life. So we can better understand this if we don't look at survival, but if we look at mortality, and we're talking about standardized age. It's very important to remove the kind of age dependentness when you're measuring the shape of life. And if you could look at the three mortality rates in the curves that I presented to you just now, they look like this. And you can see that these curves have very different shape. One of them is declining slowly, one of them is increasing quite sharply towards the end of life. So this here, for example, could be something like our Talcott Palmer or Theocrest, where most of your mortality is concentrated towards the end of life, it's very high at the end of life. This horizontal line here could be something like our rhododendron, which has a very constant mortality over its lifespan. And an important thing to note here is that we have, so I'm going to contradict what Jonathan Silvertown said earlier. I want to say senescence is not inevitable. Okay? So some things senesce and their probability of dying goes up with age. Some things don't have any senescence, they stay, their mortality is flat with age. And some things have negative senescence, so their mortality actually goes down as they get older. And there's been some research from our department which has shown that there is a big diversity of aging trajectories. In fact, senescence is not inevitable. And when I talk about shape, I'm using a measure called Keyfitz's entropy, or H. And if Keyfitz's entropy is less than 1, then you have senescence. Equal to 1, you have no senescence. And greater than 1, you have negative senescence. And actually, these should be familiar to you as the type 1, type 2, and type 3 survivorship curves that we have. But actually, what we can do with shape is we can really quantify the amount of aging that's going on there. Aging will have consequences for population dynamics, which depend on birth and death. So I'm going to talk to you about the population dynamics that we also measure from compadre. And if we look at this model, we can also represent it as a matrix. And we can put these numbers in the matrix, showing how individuals progress from one stage to another stage. And then we have the mathematical models. This is another way of visualizing what the models look like in compadre. And if we multiply that by a vector of individuals in different stages, then we get population size over time. And if the population is stable, then that rate of growth will be stable. And so the first measure of population dynamics that I'm going to talk to you about is long-term growth, lambda. And this is basically, you could say this is the fitness of the population's phenotype in a stable environment. But we also know that if you don't have a stable structure, then you might grow faster or grow slower or fluctuate in growth. And these are called transient dynamics. So I also wanted to capture the possibility for the population to have transient dynamics. And transients basically describe the population's response to disturbance. So you could have a high, you could have a disturbance which knocks you down, but then you can have transients which bring you back up, or you could have transients which actually make a disturbance worse. And we can put bounds on these transient dynamics. So we can say transient dynamics are not going to be larger than or like these fluctuations away from stable growth. And it tells you about population attenuation and population amplification. So following this disturbance, populations can attenuate, become smaller than stable. If they were stable, or they can amplify, become larger than if they were stable. And we can measure lots of different points along this, these transient bounds, and then do a principal components analysis. And what you get out of that is the first principal component which gives you the potential for transients, so how wide these transient bounds can be. And the second principal component gives you a tendency towards attenuation, so how much more is the population likely to attenuate over amplify. So I'm going to present some results on that now, and these are not published, so I've got my little no social media sticker here. And what I did is I did Bayesian markov chain multi-color generalizing in mixed models in R, looking at how these dynamics are related to the pace and shape. And this tells us something about the fitness of different aging types, and also the way different aging types respond to environmental disturbances. I controlled for phylogeny, but it wasn't important in any of the models, so these are models without phylogeny. So we can see that there's not much going on for stable growth. There's a slight and significant increase for your pace. So long-lived organisms are very slightly more likely to have a higher fitness in stable environments. This could make sense if you think about archaic selection, because they want to invest in their survival in stable environments. There's nothing really going on with shape. And what we can also do is we can plot this in a pace-shaped space. So something down here is very short-lived, but also has a, so if it's at 1, has a relatively uniform mortality across its life cycle. Down below 1, as we said earlier, is senescence, and above 1 is negative senescence. So you can also see that most populations here are negatively senescent as opposed to senescent. We get more interesting results for transients. So there's a decline in the potential for transients with increased longevity, and that again makes sense if you are going to invest in stable growth because you have long-lived high survival. There's also a decline in the potential transients for shape. So I was actually expecting there to be a curved response here. I was expecting it to be that if you had a more extreme shape, then you also had a, so in either direction, so if it's senescent or negatively senescent, you also had a more extreme transient dynamics. But that's not the case, and this could be because fecundity. And again, you can plot them in pace-shaped space. So things down here which are senescent and also short-lived tend to have a higher potential for transients. And so these are going to be things like annuals and stuff, so that kind of makes sense. And if you're longer lived and you are negatively senescent, then you have a less potential for transients. What we do see for the tendency towards attenuation here is this interesting curved relationship, so you're more likely to tend towards attenuation if you have a sharper shape, either senescently or negatively senescently. And that kind of makes sense because attenuation is governed by depth. So if you have a higher kind of inequality of depth across the life cycle, then there's parts of the life cycle which will attenuate very sharply. And again, in page-shaped space, we can see that individuals down here, so these annuals, are having a high tendency towards attenuation, probably because they have like this very sharp death part of their life cycle. So they could, if you get disturbed in the wrong way, then you could attenuate. And the other way around for individuals which have long life cycles. So the take-home messages are age patterns of death. Well, we've covered survival here with death, but also birth for amongst organisms. Aging does have consequences for population dynamics, and certain aging types tend to have certain population dynamics, and that tells us something about their fitness and also how they respond to environmental disturbances. But the residual variance is also very high, so there's lots of unexplained stuff going on in there. And it is just a preliminary analysis for a project that I really want to do next year, so any feedback or ideas are very welcome. Thank you very much for listening.
We actually have plenty of time for questions because apparently the next talk has been cancelled. So I
15 minutes of questions?
no. We don't need to do that, just in case people is really looking forward. So is there any questions? I had one question. So how frequent is negative senescence, prospective positive senescence in the...
If we're to believe these models in compadre, then it's something like 90% of them are negatively senescent, whereas only a very small proportion of them are actually senescent. But then this is just for survival. So of course there's a possibility that you could have reproductive senescence in those organisms. But in terms of survival, we've taken these survival curves and simulated them from the data we have, and the vast majority of them are negatively senescent, and so their mortality decreases with age. Yeah.
Interesting. Could you maybe just the different transient dynamics you're talking about? Is it possible for populations to flip between those, or do they tend to stay within one or the other?
It's a sort of more of a measure of overall tendency. So, and if you don't know the population structure, then you don't know what the transient dynamics will be. But these are a measure of the potential, if you like. So it's the potential for population to really attenuate if it's disturbed, or the potential for a population to really amplify. And the difference between those two things is how the population, exactly what the population structure is. So if the population structure is relative to the stable structure is biased towards very reproductive individuals, so high survival, high reproduction, then you'll get amplification. And if it's like biased towards individuals with this low reproductiveness, then you'll get attenuation. So that depends entirely on the disturbance, but this just describes the overall kind of potential for either the size of these bounds and how biased they are towards larger attenuation over amplification. So it just the short answer is it depends on the population structure.
So it can depend on the disturbance.
Yeah.
Whether it's impacting on one particular class or the other.
Exactly, yeah. So if you think like something like a hurricane which fells trees, that's going to be felling, the larger trees tend to be the ones who suffer, and they are also probably the most reproductive, although there is senescence in trees, so it depends on their age and their size. But say they're more reproductive, then you would get population attenuation, because you'd lose individuals from the population, but you'd also lose what those individuals were contributing to the population, and it would take a little bit longer to recover. It would be the other way around if it was something like a fire which killed all of the small trees. You would probably have population amplification then, so you get lots of seedlings growing back up.
questions? Okay, thank you very much. So as I said, the talk that was supposed to be now has been cancelled, so I guess we will continue, since we have to keep the schedule, in 10 minutes with the next talk. Okay.
Skeptical about what was I saying about? So I was trying. So there are a couple of things I was trying to unpack. So I was just really surprised, first of all, that there are species that live less lower longevity. That was just a real surprise. I was trying to think why. And you sort of answered it by decomposing these different components on the surface. And I guess I was trying to think which of these is actually what's driving them. And I think in your results you did answer that, but not actually explicitly. So that's probably what my face was trying to say. I don't think so. So maybe rodeo is basically like this. Is the reason because after that they don't start. Yes, that's right. So you sort of looked at both of them. We're not visual. And to me that would I would have done your thing you had your question right. It just seemed to me I was trying to understand why actually sometimes whether it's more to do with the concept
but sometimes this connection is lost as well.
I've yet to irony is that particularly it's probably your best option.
you plotted right in the beginning a figure of how you had to face and you had natural population, and you had they were real anyway, the point is that for those that have that go the wrong way, people actually compensate, and it shouldn't come out consistently in one direction or the other. Because it consistently with the light of Dover, which means that and you can do, it may be better than so, and it may be No, just one species you don't think so, you can't have another species. So you showed a number of plots where you had monitoring the population. Basically you and you plotted lines. And I was interested in studies, but I wanted to write that here. We started to look at traits like size, we didn't have so many non-native populations for the experiment. Actually, I was more pleased with error. So you know when you do a conventional aberration movement, you put x against y. And actually you're trying to minimize the error in the y. So so that assumes I'm not trying anymore Yeah, right. Okay. So actually, if you've got where you minimize the error in this always the ratio of the error basically so here it's almost so that's why I like refining. So there's and there's another one where you can actually zero return right you can specify. And I believe there's a version of it. I guess, you can confirm that is, actually, yes, but all your slopes are supposed to be a lot steeper. No, some of them, you and with your errors only in the ends, Right, exactly. It could really change the population. I'll find it for you. I have it. It's quite an old paper, but it can start. I just wondered if you could answer reproduction.
Okay, hello everybody again. We're continuing with the session, and now our next speaker is Daniel Sherman, who will be talking about ecology and anatomy derived plant demography work.
Hello, it's my first talk in a Christmas jumper, so we'll see if it goes well. I'm based at Maxo with Ian Stott at the University of Southern Denmark. I'm doing my PhD with Owen Jones and Johan Dahlgren. I'm going to be talking to you about ecology and anatomy. Anatomy of plants is kind of an odd word, so I'm sort of looking at the environment that they're growing in and their growth forms and that sort of thing. But as I work in a biodemography group, I'm going to be talking to you about trade-offs. These trade-offs represent the cost paid in currency of fitness when a beneficial change in one trait is linked to a detrimental change in another. So in other terms, so when you assume that it's just selection that drives these changes, the trade-offs would occur only as a result of their history and design. But these traits stay within limits, so that means that these trade-offs must occur. There are many different kinds of trade-offs, not just demographic ones, but in this talk I'm going to be talking only about life history trade-offs and specifically about a trade-off between reproduction and survival. And the main question is, how do these life history trade-offs differ between taxonomic group and ecoregion? So I'm going to then move on to the hypotheses I have. So the first one would be that trade-off strengths are going to be governed more by ecoregion than taxonomic proximity. And I suppose the thinking behind this is that you generally get a lot of different genera that occur in a single ecoregion, but more often than not, you don't get, was that the right way around? So you get many genera in a single ecoregion, but you don't get a single genera that only tends to occur in an ecoregion. And it follows on to the larger part of the variation in trade-off strengths is only going to occur in lower taxonomic groups. I've talked to myself in circles with this many times, so I'm hoping that this image is going to help you slightly. So if you just imagine that the black dots on the screen, and we're just going to focus from the upper half, the black dots on the screen are trait values for trade-offs. So it moves from strong to weak, from left to right. And if you assume that the orange boxes are families, so lower down in taxonomic hierarchy, and the blue boxes are, say, for example, classes, so higher up in the taxonomic hierarchy there. This isn't phylogenetic related. I will come back to that later on. So we're just talking purely hierarchical taxonomy. So you're assuming that the variation, the differences in trade-off traits happen lower down. So that's the first example. So the variation is very neatly explained by a lower taxonomic hierarchy. But in the example at the bottom, there are many more differences. The thing to look at here is the blue boxes, that they're more disorganized, so that the variation in these, the trait strengths of the trade-off happen at a higher level. And last but not least, trade-off strength varies systematically with ecoregion with stronger trade-offs in nutrient-poor environments. So you're assuming the more stressful the environment, the stronger the trade-off's going to be in that area. As Ian explained to you in the last talk, I'm working with Compadre as well. It's a plant matrix database. It contains 695 unique species. I have not used all of them. I've limited the number of species I've used to ones where there are actually more than 5 matrix projection models already present in Compadre. I've also only used matrix models that include things only the active stage of the plant, so nothing that includes a seed bank. Seed bank issues will have issues for fertility later on, but I'll come back to that. So this is only things that have an active stage class. So I had to generate a quantitative measure of this trade-off, and I did that by using the weighted mean of fecundity and survival from each matrix of each species. And the mean of fecundity was weighted by the stable stage distribution of each of the matrices. And I used the two models, so it's survival and lambda on its own, to correct for the population growth rate of each of these matrices. Because in my analysis at this point, I haven't included any effects of where the data has come from different populations or different years. So I'm attempting to correct for that slightly by correcting for lambda within the model. The slope values of these were then used to quantify the trade-off. So if we go back to the first hypothesis, you can see at the bottom, so this box plot has the orders along the x-axis, and it's been subsetted to the orders with the most amount of data. There were several other orders, but there wasn't enough data to show anything that would be worth it here, and the value of trade-off is on the y-axis. And because it was sloped and you wanted to see a trade-off, a negative value means that there was a trade-off there, but its significance is still yet to be determined. But you can see here that it's more likely to be the family, so it's more likely to be the taxonomic hierarchy that explains the variance in the trade-off value. So a larger part of variation in trade-off strengths occur in the lower taxonomic groups. From the variance values, you can see that it's clearly family that explains the variance and not the other taxonomic hierarchies. And the next most variant thing is the ecoregion. So it is the lower down you go, the more likely you are to be able to explain the trade-off. So essentially what you want to get from this is that how far down in the hierarchy do you have to go to be able to say, I know kind of what the extent of this trade-off would be. So if you, in this case, if I only gave you order or class information, you wouldn't be able to tell the information you'd need as family. This isn't my results. This is just what would be the ideal thing to see if you wanted to see a very clear distinction between the different trade-offs varying systematically with ecoregion. This is a very ideal data set, but we live in the real world, so it looks like that. Which you can't really see much differences in a lot of them, but I suppose the most important thing to look at here is that desert seems to have a much stronger trade-off than any of the other ecoregions. And so you could say that the desert, in this case, is the most nutrient poor and the most stressful environment for the plants, which leads to a stronger trade-off. So overall, I guess no for the first one. Trade-off strengths tend to be governed more by taxonomic proximity. The trade-off variations are lower down at the family level. And the last one, yes, it does-ish, so in a more stressful environment like deserts, you do seem to see a stronger trade-off, but it's kind of hard to distinguish between the others at what level this trade-off is. But following on from this, I would like to include seed banks, because I think it's a really important reproductive thing to include. And I promised I'd get back to phylogeny. I'm sorry if you feel duped, it's only in the bit that I haven't done yet. But phylogeny is an important thing to include because it distinguishes at what point the species diverge. So you haven't included the time element when you just talk about taxonomy, and I think including that time in the point at which the species diverged is really important in determining whether it is actually their phylogeny or it's more ecoregion. So that I'd need to include that and then go back to the first hypothesis about taxonomy or ecoregion. And growth, I want to include clonality. I've so far just looked at a binary value for clonality and found there wasn't a difference between plants that were clonal or not. But quantifying the trade-off, I'd like to quantify essentially how clonal a plant is. I attempt to do that and try again and see if there's anything different. and growth form. I made this brief preliminary analysis, and most of the growth forms tend to be quite around the same value, but it's the epiphytes that are the interesting ones that seem to show less of a strong trade-off, but they're also the only growth form here that tends to live on other plants. And that requires more investigation. But for now, thank you, and I'd like to thank the Max Planck Society for my funding, and my supervisors, Owen and Johan, and Ian, and the Compadre team, and to all the people who gave data to Compadre that allowed me to do the analysis. Thank you.
Could you tell me what your opinion about this? You are using Compadre, no?
Yeah.
And it's fine. However, Compadre does not represent a natural sample of the diversity. It's a biased sample of diversity.
Yeah.
How much do you think your results are also biased for the membership of Compadre? If you could really collect a sample from the diversity,
Yeah.
the result would be the same?
I suppose the truthful answer is I don't know. I know, for example, that in the tropical forests part, like ecoregion of Compadre, I think there's a larger amount of tree data than there is any other species. So I would assume that yes, that would have an effect, but I don't know what that effect would be at this point in time. But I suppose that's partly where I want to include growth form into this analysis, because then you could get a more representative idea of what each of the types are doing, and then I will become more aware of where more of the gaps in Compadre's data is, in terms of the analysis I'm doing anyway.
So does this database have information on genus and species as well as family? I'm wondering why you didn't analyze at those levels.
Yes, it does, but I suppose that was an error in my own analysis, because the way it's broken down is it does have them individually, but I had grouped things by entire species name, so genus and species. But yes, I should probably run it again with genus and species as variables as well.
You may run into issues with sample sizes at that point.
Yeah.
Just at the end there, that was interesting that you were showing that maybe epiphytes have fewer trade-offs there.
Yeah.
Do you know if in Compadre epiphytes are there species in there that are parasitic or symbiotic? You know, that maybe the epiphytes are getting growth or nutrients from the plant they're growing on, and that's why they're so strong trade-offs.
I'm sure that information is in there. I haven't looked for it, but yeah, absolutely.
So, can you clarify a little bit how were you, when you were analyzing the trade-offs, you were analyzing which processes were you, survival, reproduction?
Yes.
So you were using the matrix elements?
So I calculated the stable stage distribution of each of the matrices, but divided matrices only. So I took the individual matrix U and matrix F. So this was just for sexually reproducing plants, and I got the stable stage distribution from those and calculated a weighted mean of the fertility matrix and a weighted mean for the survival matrix, and that's what I used for those values in the model.
Okay.
I'm just trying to put what you've shown us into other words. So can I just try to
Sure.
say, maybe totally wrong, but I wonder whether what you're telling us in your trade-off strength has to do with arraying species along a fast-slow axis, because fast will have low survival and high reproduction, and conversely. And so are you saying, for instance, in the deserts, that the fast-slow continuum is more evident in the deserts than in some of the other ecoregions?
Oh. I don't know. I mean, I suppose that off the top of my head, but yeah, I think so.
Is there any questions? Yeah, go ahead.
Such a nice question. What was in the title? Anatomy?
Yeah. If anyone has any suggestions, it was sort of the word doesn't quite fit everything I'm doing here, but I needed something in the title to suggest that I wasn't just looking at ecology in terms of the plant demography. It was perhaps a poor choice of word, but it was all I could come up with at the time.
Right. Thank you. So our next speaker, yeah, where is Victor Ranget? Yeah. So he will be talking about body mass and predictions on early life survival. Sorry, yeah.
So hello, so I will present you the work on meta-analysis about the relationship between juvenile survival and Burly body mass. So I want first to thank all my collaborators on this study. So what we know is that there is a strong variation in juvenile survival among juveniles in vertebrate populations. And what we want to know in population dynamics is how to describe this variation. And one of the potential drivers of this variation in survival is the body mass. So what is the basic prediction of the literature is that body mass is positively correlated to the body reserves, and also that body reserves can enhance the juvenile survival. So the prediction is that we expect a positive relationship between body mass and juvenile survival. So if we look closely in the literature, what we see is that this prediction is confirmed in many cases. So here I see 4 examples in birds and mammals of a positive relationship between early survival and body mass. So what we see here is that we have 4 positive relationships, but we have also differences in the slope of the relationship. But also what we see if we look closely in the literature is that there is some study case in which there is no detectable relationship between juvenile survival and body mass. So the aim of this work was to draw general patterns about the relationship between body mass and early survival, and to see if there are general patterns toward our predictions that there is a positive relationship. So to do this, I perform phylogenetically corrected meta-analysis. And after, what we see is that there is different intensity in the relationship. In some relationship, we see that there is a strong relationship between juvenile survival and body mass, and in some relationship, there is no relationship at all. So what we want to do is to know the factors that can influence the difference in intensity in the relationship. So for this, I perform different meta-analysis with the same data, but with testing some fixed factors. Okay, so I will briefly comment my procedure for the meta-analysis. So first, for the literature survey, I use keywords in Web of Science. For the data reported, we reported the data for the identification of the study, the effect size, which is the slope of the relationship, and also the standard error associated. So I report also the species and the localization. So for the factors that can influence the relationship, I reported if the population is subject to predation or not, and also if the population is captive or not. So I reported only this for mammals because I have only this all the information in my study for mammals. I report also the timing of mass measurement, if it's at birth or at weaning, and also the mating system, the sex of the offspring, and the average litter size. So briefly about the statistical analysis. So I performed a multi-level analysis because my studies are more likely to be not independent because of the phylogenetic history. So I correct for the phylogeny. Because of the species independently of the phylogeny, so individuals from the same species can share the same characteristics which are not dependent of the phylogeny. I correct also for the populations and also for the author effect. So all these corrections, I use them as random factors in my model. So for my first question about the general patterns, I first fit a general model without any fixed effect. So I will present you the meta-analysis mean, and I'll also perform an heterogeneity analysis to show the proportion of variance explained by each of my random effects. And also I test for the publication bias to see if there is a lack of not statistically significant results published in the literature, which are likely to bias my analysis. And after, to show the factors that can influence the relationship, I included moderators, so to see the influence of the factors. For example, for the predation, I formed two groups, one in which there is predation and one with no predation, and I compare the mean of the two groups. So about the data set, so here is the keywords I use for the meta-analysis. So we ended up with more than 20,000 papers. So from all these papers, we select them based on the title and the abstract. So we ended up with more than 100 papers on mammals and on birds. And after, we can only include 60 papers in mammal and 57 papers in birds because we need to have all the data needed for the analysis. Here is all the phylogeny of all the species we included in the analysis. So what we see is that we have a large spawning of the different groups in mammals and in birds, but especially this is focused on the species in which we have a long-term monitoring, such as the Artiodactyla or the Passeriform in birds. So now about the results of the meta-analysis. So what we see here is in terms of odd ratio. So if the odd ratio is higher than 1, it means that there is a positive effect. So what we see here in mammals and in birds is that there is a positive relationship between juvenile mass and juvenile survival. And if we look closely at if there is a difference between mammals and birds, what we see is that it seems like in birds, the meta-analysis means is lower than in mammals. This is reported in the literature that in birds, the data are most likely to the juvenile survival is most likely to be not so much influenced by body mass, because, for example, body mass can be costly in terms of flight performances, and they can have, for example, in high predation conditions, issue to avoid the predators. Okay, about the heterogeneity analysis, so this shows the proportion explained by each of my random factors. So what we see is that all the mean are lower than 50%. So none of my random factors explains a high proportion of variance. So there is no effect of the phylogeny, And if we test after for the publication bias, which I've not presented yet, but I tested that, and there is no impact of this publication bias on my results. Now, if we looked at the effect of predations, we see a very interesting pattern here in mammals that in the population in which the individuals are subject to predations, we see that there is a negative effect of the predations on the meta-analysis mean. It means that there is in the population in which the individuals are dying from predation, there is less condition dependence. Also, what we see is that there is an effect of the captivity on the intensity of the relationship. So we see that there is a higher meta-analysis means for the captive one. So in captivity, there is a higher condition dependence than in wild. And if after we test from the other non-environmental factors, such as the timing of mass measurement, the litter size, the mating system, and the offspring sex, and what we see is that there is no effect at all of all these factors. So what are the conclusions of this, of all the analysis of all these factors? The conclusion is that this relationship is likely to be very dependent of the environmental conditions. So what we see is that when the individuals are dying from predation, there is less condition dependence. So it is likely that the relationship between body mass and the probability to dying from predations is not so high as the relationship between body mass and starvation. And if we come back to our predictions that the body reserves can enhance survival, it can be also that the body reserves enhance survival only in case of starvation, in case there is food limitation. But in predations, body reserve is not impactful on the survival. So what is the take-home message of my presentation? So the take-home message is that there is a positive pattern towards a relationship between juvenile survival and juvenile body mass. And what about the difference in intensity in the relationship is that this is very environmental dependent and dependent of the condition. And especially to have a clearer view of this relationship, we need to know how body mass can affect the probability to die from predation, starvation, or diseases, which are the main causes of mortality in wild. So this is an open question. What is really the relationship between body mass and the cause-specific mortality? Because we know from our results that it is likely that body mass is not related to predation, but when I take into account the predations, I only take into account if this is subjected to predation or not, but I not take into account the proportion of individuals dying from predation. Is this is an high proportion of the population or not a high proportion? So we need to do that to have a clearer view of this relationship. So thank you for your attention.
I'd like to ask in that next presentation, have you divided between chick survival and juvenile survival in birds, or have you used only juvenile survival after fledging?
I have tried to test this if there is if the survival is tested at fledging or after, for example at recruitment, because I take all the relationship between birth and recruitment, and there is no effect at all in birds and in mammals. But
only fledging and juveniles after fledging used in your analysis?
Yes.
Perfect. Thank you very much.
Any other question? Yes.
So bigger bodies, it's not just bigger fat reserves, they may be structurally bigger. So you're suggesting that bigger bodies are good because they have more fat reserves. Have you actually got any evidence that there are more fat reserves? Because my impression is, which may be completely wrong, is that there's such enormous selection pressure to grow to maturity fast that a lot of species, the juveniles, don't put anything aside as fat reserves.
We try to use other indices as body mass. For example, body condition, which is more likely to be, which is mass corrected by size, so which is more likely to be related to reserves, and there is the same effect for body condition and body mass, the same kind.
Thank you very much. Thank you. Our next speaker is Svenia Kroger, who will be talking about cumulative reproductive costs on current reproduction in a wild mammal.
Yes. Does this work?
Oops. Just one second.
Yay.
Okay.
Thank you. Reproduction is costly, but does breeding again and again and again determine whether you can breed again? Or is it mostly what you did in the previous year that determines what you can do in the current year? So these are some of the questions that I'm interested in, although I'm also a student of senescence, actually. But today I'm going to talk to you about cumulative reproductive costs in a wild population of yellow-bellied marmots. Individuals have limited resources they can allocate among major life history traits, such as growth, survival, and reproduction. And as far as reproduction is concerned, individuals also have to balance how they allocate resources among current versus future reproduction. Because the idea is, if they allocate more resources to current reproduction, they'll have fewer resources to allocate to future reproduction. Now, what some people have looked at is how previous year reproduction affects current year reproduction, and usually if there's a so-called short-term cost of reproduction, you'd expect that an individual that bred in one year might not be as likely to breed in the subsequent year. However, it could be that what you did in the last year doesn't really affect what you do in the current year in terms of reproduction, but then, if you look at the whole previous reproductive lifespan and add up all the reproductive events, that could have an effect on current year reproduction potentially. Then we can add another layer of complexity to this, because deer, and in fact many species in which people have looked at cumulative reproductive costs, are monoticous species, which means they give birth to a single offspring, or sometimes twins, per reproductive event. Whereas marmots, for example, they can give birth to litters up to 10 pups. So then the question is not only did you breed, but also if you bred, how many offspring did you have? And both the fact that you bred and the number of offspring you had could affect whether or not you're able to breed in the current year, and also how many offspring you have if you do breed. And this is sort of an overview of what I'm going to talk to you about today. And as I said, not a lot of people have looked at cumulative reproductive costs, especially not in a species with high variation in litter size. Now this leads on to my questions, which are, what are the costs of previous reproduction on current reproduction? And I'm not only going to look at short-term, but also cumulative long-term effects. I do this in the yellow-bellied marmot, very beautiful creatures. They're long-lived, long-lived up to 14 years in females, hibernating mammals, rodents, that live in the Rocky Mountains in Colorado at about 3,000 meters altitude. And this is a long-term study that's been going on since 1962, so we know a lot about these guys. For example, we know their exact age, because we captured them as soon as they emerged from the maternal burrows in spring. And we give them these uniquely numbered metal ear tags upon first capture, so we can follow them throughout their lives and we know their life history. Consequently, we know their reproduction, so we know when the pups come out of the burrows and we know who their mother is, because we don't just trap them, we also observe colonies extensively. Now, to get to the models, I'm really only going to scratch the surface of this, but I'm very happy to chat to you afterwards if you want to come and find me. But what you need to know is that my response variables in the models I used were, first of all, current breeding probability. This was a generalized linear mixed model with a binomial distribution. And then I had another set of models, and I'm going to talk about those in terms of the number of pups a female had if she did reproduce. And in my data set, that was between 1 and 9 pups. And this was a linear mixed model with a Gaussian distribution. Now, the way in which I measured previous short-term reproduction was I looked at whether or not a female bred in the previous year. And I looked at how this affected her current breeding probability or litter size, respectively. Then, in terms of previous cumulative reproduction, I used 2 different measures. First of all, I looked at previous breeding frequency. And now this is a continuous variable, but for the illustration purposes in this talk, I have also given it the categories of high and low, and you'll see in a moment what I mean. And the breeding frequency is essentially a measure of how often she's been breeding since she's been reproductively active. And then I have another measure of called average litter size, which is in essence a measure of how many total pups she's had in the past. Now, if there is a long-term cumulative cost of reproduction, what you'd expect to see is that females with either high previous breeding frequencies and or many pups in the past would be either less likely to breed again in the current year or maybe have lower litter sizes if they do breed. And now we move on to the results, and I'm also giving you my sample sizes here for the current breeding probability and litter size models. So what did we find? First of all, in terms of short-term effects, on current breeding probability, nothing. Whether or not they were breeding in the previous year did not affect whether or not they're bred in the current year. Same is true for current litter size, so whether or not they're bred did not seem to affect litter size in the current year. So in terms of short-term effects, there was nothing in the variables that I looked at. Then what about long-term effects? First of all, long-term effects on current breeding probability. The breeding frequency had no effect on current breeding probability. Neither had previous average litter size, the number of pups they've had in the past. However, now it gets interesting, because if you take the interaction of the 2 previous long-term cumulative variables, that's when we get this, and that's when things get exciting. So here I show you the current breeding probability on the z-axis, and on the x and y-axis you have the 2 previous cumulative long-term variables of previous average litter sizes and previous breeding frequency. The 3D surface shows you the model predictions of the generalized linear mixed model. And the colors indicate the values of current breeding probability. So the red shades essentially indicate lower current breeding probabilities, and the yellow shades indicate higher current breeding probabilities, and if they're lighter, they're higher. Although it's a bit difficult to make out some of the shades on this graph, I'll admit. Now you'll notice that, I should say the points represent the distribution of the raw data. You'll notice that in this parameter space I've circled here, there's no data. And actually this makes perfect sense. So these are individuals that on average have had very low previous breeding frequencies and really high litter sizes. This means, well, it's very unlikely that individuals that have hardly bred at all will have had the highest total number of pups in the past. So this doesn't make a lot of sense biologically, hence there is no data. Now, if I can draw your attention to this bit, this shows that essentially individuals with the lowest previous breeding frequencies and the fewest pups in the past on average have got low current breeding probabilities on average. So they're just doing persistently badly, according to this model. Then you can see a lot of observations are kind of on this saddle, they're kind of intermediate values. But individuals which had high previous breeding frequencies but small litter sizes, on average, had quite high current breeding probabilities. But now, when you have high values for both frequencies and litter sizes in the past, that's when current breeding probability goes down, suggesting that there might be a cost involved if you have too much of everything. What about the effect on current litter sizes? Well, first of all, again, there was no effect of just the breeding frequency on current litter size. But individuals which on average had higher litter sizes in the past also had higher litter sizes in the current year, which I'll get back to briefly in a moment. Then this graph essentially shows the same thing as the previous one, but this time you have current litter size on the z-axis. And The same parameter space has no data due to the same reasons I've mentioned before. Again, individuals which have low values that haven't reproduced often in the past and had small litter sizes also have current litter sizes, so these poor guys just always do bad if they're there, on average. And then this time, individuals which had high frequencies and low litter sizes have sort of intermediate values on average. And this time, individuals that did a lot of everything, high frequencies and high litter sizes, still did pretty well in terms of current litter sizes on average. Now, what does this all mean? First of all, I did not find any evidence for short-term effects, so nothing suggested that there were any short-term costs of reproduction, which is really interesting because a lot of studies have actually looked at this and didn't find anything, and it might be worth revisiting them if there's the data, which of course a lot of people don't have such high-resolution data on so many years. But besides persistent among individual variation in litter size, I also found that there were some long-term effects that suggested that there might be a cumulative cost of reproduction in individuals that had, on average, high breeding frequencies and high litter sizes, so a lot of everything. So this reduced current breeding probability, but not current litter size. And what does this mean? This is really interesting, because this shows that potentially because if really individuals with high frequencies and high litter sizes did have reduced breeding probability, if they do breed, they have big litters, this either suggests that these individuals are really awesome, so high quality, although I hate this word, or it could be that they transfer the cost to their offspring. So this is something I'm very interested in and looking at the moment, because it could be that those pups in those big litters might just be performing really badly and consequently reduce the lifetime reproductive success of the mother, which of course I can't see in these analyses, but I'm hoping to look at the moment. So that only leaves me to thank my supervisors, Julien Martin and Jane Reed, and all the people involved in this project, especially all the many marmoteers who collected data since 1962. And then I'll thank you, and I'll be happy to answer any questions.
And it's not possible that if these are social animals, actually it's like in rural areas in Mexico, like the more children you have, the more labor you have, and the better the family overall will do under those conditions, no? Obviously other circumstances will result in other things, but that helps, no?
Yeah, it is a social animal indeed. And there's a lot of studies going on about this. I'm not really sure. I didn't test in this study. I didn't look at how social interactions or the social network affected their breeding success or anything else that I've looked at. But it can sometimes have advantages. It can have disadvantages. I think it would be really interesting to look at that as well, actually. But I don't know if I'll have the time. I'm close to finishing. So but yeah, it's a very fair comment. Yes?
Over the many decades, you've got very variable years, and different females breeding in those different years may be impacted by that. So I wasn't aware in this study that you factored in the environmental variation, which could be producing some of that overall maternal individual difference.
Yes, that is a very good point. So in fact, obviously I didn't talk about my models very much in detail. I did look at some environmental effects as well. So for one, I did have random year effects in the models, but that of course doesn't account for everything. But also, I think to look at some of these effects, my data would probably not be powerful enough. However, I have looked at some environmental effects in terms of space, and they seem to suggest that depending on the exact part of the environment that females were living in, there was more likely to be a cumulative cost at, for example, slightly higher altitude. Reproducing more often seemed to reduce their probability to breed again. So there's definitely something environmental going on, and I have some evidence for that, but in the interest of time, I wasn't able to present all of that. But that's a very good point, yes. Yes?
I'm curious to know what would be the impact of traits such as age or body mass in relation to the previous talk where body mass could reflect the amount of body reserves that you have.
Yes. Yeah, that is also a very good comment. Also things I have looked at. So in terms of body mass, of course in marmots it's very important because their body mass between the active and the hibernating season varies up to 40%, so that is a lot. And in fact, it's a bit of a vicious circle because reproduction affects body mass, but body mass affects reproduction. So in essence, body mass is kind of the underlying mechanism of this. So it was not included in these models, although I have looked at it, and it didn't cause any problems. So yeah, in relation to age, I also looked at age effects and, for example, tested for interactions between the previous reproduction variables and age, because it could be that maybe only older individuals have these cumulative effects. And that would also actually be getting at disposable soma theory, if you're at all familiar with that. And again, there was no evidence that that was important in the analysis I did. So, but yeah, very good points indeed.
In the short-term results, was there an effect of the interaction of whether or not they bred last year and litter size just from the previous year? So I know you said there was no effect of either the interaction. Did that show anything, or was that something to look at as well?
Sorry, do you mean the interaction between breeding the previous year and the litter size the previous year?
If there was any short-term effect, because you saw the interaction effect kind of from the cumulative, so I just wondering if there is any slight effect short-term for the interaction as well.
No, but I also, no, there was nothing of the sort.
Okay, that's
Okay.
Thank you.
Thank you.
And our today's last speaker, Hannah Freud, she will talk about whether age-related variation in space use contribute to demographic census.
Oh, I can do that. Don't worry. Where is the pointer? Where's the pointer now? Oh, here it is. There you are. Okay.
How do I get the pointer? Is that, or is it this one?
Yeah, that's the pointer.
Oh yeah, perfect. Yeah. Sorry. Thank you very much. Okay. Hello. Thank you all for making it here to the graveyard shift of the conference. I'll try not to keep you for too long. My name is Hannah Freud, and I work as a postdoc in Edinburgh for Dan Nussey. I'm just going to talk a little bit today about an ongoing project we've got looking at space use into unmanaged populations of ungulates in collaboration with Charlotte, a PhD student in Edinburgh, Josephine, who runs the long-term studies, and Luca, who works in Swansea. Today, yeah, sorry. So the long-term projects that I'm going to be talking about are the Sowie sheep of St Kilda and the red deer on the Isle of Rum. So despite kind of early skepticism about the idea, it's increasingly being shown that demographic senescence does occur in wild animal populations, so declines in reproductive performance or survival with age. But what's going on with other traits is a little bit less clear. And in particular, what's going on with behavioral traits is particularly not very well documented. So, of course, we know that behavior is really highly variable, both between individuals and within individuals, and it can be quite hard to characterize. But we know from kind of studies of humans and domestic and lab animals that a number of kind of physiological functions that are known to decline with in later life, like things like athletic ability, visual acuity, we kind of expect from first principles that they're quite likely to affect behaviors, particularly things like foraging behavior, which of course might be really important for fitness. So there is a possibility that age-related changes in behavior may underpin some of the demographic senescence we observe in these populations. And there have been a handful of studies have looked at this, some really interesting stuff on like wolves and moose, but actually particularly recently there's been this really massive increase in technological advances, and people are now really interested in biologging and tracking, so kind of GPS tracking using accelerometers, time depth recorders, things like that, which give really, really amazing high detailed information on individuals over quite short periods, so maybe a few foraging trips or whatever. And this really, really high resolution data
is amazing for those few foraging trips, but often it's quite low in terms of coverage. So we might be talking about just a handful of individuals or just over quite a limited time frame. And when we're thinking about long-lived individuals, there's a wonder about whether looking at cross-sectional patterns in these kind of short time snaps is representative of what's going on for the individual throughout their whole life. So we're taking a bit of a different approach, and we're going, instead of for very high resolution, we've got quite low resolution data, but covering over the whole lifespan of many individuals. So we use data from these long-term field studies, sheep on St Kilda and red deer on the Isle of Rum. So these populations have been monitored for many decades now. All the individuals in the populations are marked at birth, and we have really amazing life history data for individuals following their whole lifespans. And importantly, demographic senescence is evident in these populations, and the patterns tend to be quite sex-specific, so there's really strong sexual selection in both populations, and this has consequences for kind of the patterns we observe. So it's kind of important to consider the sexes separately in analyses of senescence. So the data we use from these studies is census data. So our wonderful field assistants, so 30 to 40 times a year, depending on the system, they walk a set census route, and they record the location of all individuals that they observe to 100 meters. These census are taken place throughout the year, so kind of a number of censuses per month, and kind of scattered through various different important events in the life cycle of the annual kind of year of the sheep or deer. And we now have, over the number of decades that this study's been running, kind of hundreds of thousands of observations of individuals over many decades. So we wanted to use these observations to generate annual home range estimates using kernel density methods. So then we wanted to look at how some aspects of these home ranges varied with age. So firstly, considering just how annual home range size varied with age. And also in the sheep, we are lucky enough to have some information on vegetation cover. And previously, Charlotte has done some work looking at how proportion of holcus, kind of vegetation community across a home range, can be associated with kind of increases in lifetime reproductive success. So we have a kind of metric of home range quality for the sheep as well, and look at how that may change with age. The first problem that we kind of encountered was that individuals vary in the number of observations that they had. Sorry, it's quite small, but just a number of observations against the average home range area. And you can see that there's quite a strong negative relationship. So more observations tend to result in a smaller home range estimate. And that's partly kind of just a statistical artifact, just to do with the way in which the kernels are estimated. But it also potentially has some biology in there. So, for example, individuals are in certain locations. They might be more likely to be observed more frequently, but they also might live in kind of better quality habitats and generally maybe have smaller home range areas. So we wanted to control for that variation. So we just decided to subsample our data using a kind of structure. So we sampled kind of throughout the year, and so just to equalize the number of observations for all kind of individual years, so we can control for that sampling bias. When thinking about the age effects, we, of course, when we are looking at across the age range, the number of individuals, this is just a plot for the deer, but the number of individuals that are surviving until the oldest ages is obviously kind of quite small. So this bunch of individuals down here are actually probably not a random subset of the population. There is a reason that they are the longest lived. And actually the selective disappearance of individuals kind of in successive age classes can lead to a generation of patterns at the population level when we're looking at how average trait values change with age. So we just included longevity and its squared term in our model to account for the kind of selective disappearance of individuals in our population. And that means when we include age and age squared in our models, we're looking at within individual aging effects, which is what we're interested in if we're thinking about senescence. We also included a factor for final year. So just in case the patterns are kind of not gradual. For example, in the sheep, we see kind of a terminal decline in body mass. So individuals in their last year of life tend to lose quite a lot of weight. So just seeing whether we can pinpoint some of that to changes in home range. Aspects of home range. We included some additional fixed effects and also random effects of year in individual. So this plot is just, in all of the plots I'm just showing raw data averages and standard errors. And here we've got age on the x-axis, and home range area on the Y. This is deer home ranges, and it's measured in hectares, and I've logged it just to normalize it. We've got boys shown in, it's blue, but I guess it kind of looks green. So it seems like for males, we find a quadratic relationship between age and home range area. So the youngest and oldest males tend to, when they're younger and older, have larger home ranges than kind of prime age males. Thanks. And with females shown in red, it kind of looks like perhaps a similar pattern, but actually when we include longevity in the model, it's only the linear age term that's significant, so a significant decline in home range area with age. And that's because we find a significant association between average home range area and longevity in females, and that's actually a quadratic relationship. There's no relationship at all in males, but in females, these kind of shorter-lived individuals tend to have larger average home ranges. So their selective disappearance from the population kind of in earlier ages contributes somewhat to the successive decline in home range areas that we see at the population level. But then these kind of longer-lived individuals, with much smaller home ranges, as they're successively lost from the population, we then see a kind of increase when we're left with just these individuals that are quite long-lived and actually have larger than average home ranges. When we move on to look at the sheep, again, we see kind of different patterns between the sexes. Females, for sure, show quite a strong decline in home range area as they get older. Males, perhaps some suggestive increase, but I'm not going to put too much emphasis on that, but still something slightly different anyway. And in the sheep, we actually find no relationship between average home range area and longevity. So what's happening here at the population level is quite representative of what's going on within individuals. So I guess particularly with the females, we're seeing these kind of declines in home range area as they get older. I guess tentatively we could start thinking, well, maybe that's kind of some reflection of mobility. Maybe they're just moving around less as they get older. But in the sheep, we have this data on habitat. Oh, sorry. Sorry, I just missed a final thing. Actually, when I include a factor for final year, we actually see a terminal decline in home range area in both males and females in the sheep. So that kind of mirrors that relationship with body mass that I mentioned. Sorry, getting ahead of myself. So when we look at home range quality, in female sheep, we actually see an increase in home range quality with age. And in males, again, we see nothing going on. This is independent of the fact that home ranges are actually declining in size as we get older in the sheep. So we have this negative relationship between home range quality and home range area. So larger home ranges tend to be of poorer quality, but this is significant independent of that. And also independent of the fact that females with better quality home ranges tend to be longer lived. So there is some contribution of selective disappearance of females with poor home ranges, but we still see this within individual increase in home range quality, right into the later stages, is still significant. So just to summarize, we detected some selective effects on our population-level patterns of home range area and quality, but we also do see within individual declines in home range size in females, both in the sheep and the deer. We initially wondered whether this could be indicative of some kind of senescence, but particularly in the sheep, I don't really think that's the case. So we see that home range quality actually improves with age in the sheep. Perhaps this is some kind of effective experience. They just know where the better areas are, so they're able to just kind of winnow in them. Perhaps also there's some kind of social dominance thing going on, so maybe older females are more dominant and they're more able to kind of dictate their foraging areas. We do see these terminal declines in home range area, so it would be interesting to put those into a model of body mass and see if we can explain some of the final decrease in body mass with declines in home range area. I haven't got to that yet. And also in the deer, I'm quite interested to see whether the scales that we're talking about here are quite different. So in terms of the sheep, their home range areas are just much smaller on average, but in the deer, they're quite a lot bigger. It seems that with sheep, we have this information on home range quality, and it's not kind of indicative of senescence, but I would be quite interested still just to have a look with the deer to see if I can put these into some models of reproductive performance or whatever, and see if the age-related declines we see in home range area maybe can explain some of that. But that's coming next. So finally, just to say thanks really to everybody who's involved in the long-term field studies, because obviously without them this work wouldn't be possible. And thank you very much for listening.
nice talk. I'm interested in this issue with the home range size declining with observation. So would it be useful in your models to actually fit the number of observations as occur there?
Yes, I have tried alternative approaches, yeah, and basically you get the same, similar results. And actually I was really worried that the correlations actually between we get, particularly with the deer, for if you just use all of the available data for an individual and this kind of subsampled data, it's actually kind of like 0.9 or something.
Okay.
So actually they're quite similar.
The subsampling gave you the same number of points for each.
Yes, exactly. So all individuals had, I had say 12 observations per year, so we're completely controlled for that. I was just a bit worried there was kind of complicated combinations of statistical artifacts and biology going on that I wasn't sure. So I wanted to be sure, but I think the full data set is actually kind of pretty okay to use, but this is subsample data.
So following on from that, why are there lower numbers from some individuals? Because if they're not seen, is there not a danger that your subsampling is not representative, that you're actually missing animals that have gone off on some excursion and you've just missed them because they're in a difficult spot to see?
Yes, I guess so. I guess some of them just live on further away potentially from the census route, so they're just kind of harder to observe, I guess. So sometimes they're just missed, whereas some individuals live right outside the cottage, and if they're there, you see them. So I guess there is some of that, but with the subsampling, I was only including individuals that were seen a certain number of times in a year, so they had to have been observed kind of 12 times in a year. So I guess we might lose some kind of peripheral individuals in that way, but I'm hoping that should control for that.
It'd be nice to stick a few tags on the test.
Yeah, it'd be nice. See how the powers that be think about it.
Okay, well, this session has been really interesting, so let's thank again all our speakers. Sometimes they watch a discussion with their count data and we are because, okay, so you, that would need to use GD